Today, we are diving into an important topic in the pharmaceutical industry, cleaning validation. Ensuring that equipment and surfaces are properly cleaned is crucial for maintaining product quality and preventing cross-contamination. So, let's explore the key steps involved in the cleaning validation process. Step 1. Defining the scope. The first step is to identify the equipment, surfaces, and utensils that require validation. This helps us focus our efforts and resources effectively. Step 2. Developing a cleaning validation plan. A comprehensive plan outlines our objectives, acceptance criteria, sampling methods, analytical techniques, and documentation requirements. It provides a roadmap for the entire validation process. Step 3. Selecting worst-case products. We need to identify the most challenging or difficult to clean products that will be used as worst-case scenarios during the validation study. This ensures that our cleaning procedures can effectively remove residues from even the toughest cases. Step 4. Establishing analytical methods. We select appropriate analytical methods to detect residues or contaminants. Techniques such as HPLC, GC, or specific microbial tests are used depending on the nature of the equipment or surface being validated. Step 5. Executing the cleaning procedure. We follow standard operating procedures for cleaning the equipment or surfaces. It's essential that our cleaning process is representative of routine practices in the manufacturing facility. Step 6. Sample collection. Immediately after cleaning, we collect samples from critical surfaces or equipment. Swabbing, rinsing, or direct surface sampling techniques are used to gather samples for further analysis. Step 7. Analyzing samples. The collected samples are subjected to rigorous analytical testing using the selected methods. We analyze the samples for the presence of any residues or contaminants to ensure they meet the defined acceptance criteria. Step 8. Evaluating results. We compare the analytical results against the predetermined acceptance criteria. If the results fall within the limits, the cleaning process is considered validated. If not, corrective actions and revalidation may be necessary. Step 9. Documenting and reporting. All steps, results, and findings of the cleaning validation study are thoroughly documented. We prepare a detailed report summarizing the validation process, analytical data, and conclusions. Step 10. Ongoing monitoring. A robust cleaning verification program is implemented to periodically assess the effectiveness of the cleaning process. Regular reviews and updates ensure compliance with current regulations and industry standards. By following these steps, pharmaceutical companies can ensure that their equipment and surfaces are properly cleaned, minimizing the risk of cross-contamination and maintaining product quality. Like the video and share it with your friends and colleagues. In the context of cleaning validation, selecting worst-case scenarios is an important step to ensure that the cleaning process effectively removes residues from the most challenging or difficult to clean products or surfaces. Here are some key considerations for worst-case selection in cleaning validation. Product variability. Consider products that represent the broadest range of formulations, including those with different physical and chemical properties. This ensures that the cleaning procedure is robust enough to handle various product types. High potency or toxicity. Identify products that are highly potent or toxic, as these pose a higher risk of cross-contamination if not thoroughly cleaned. These products typically have lower allowable residue limits, requiring a stringent cleaning process. Solubility and stability. Products with low solubility or stability may leave more challenging residues. Include products with different solubility profiles or those prone to degradation or forming tenacious residues. Equipment and surface complexity. Consider equipment or surfaces that are difficult to clean due to their design, construction, or material. This includes intricate parts, dead spaces, crevices, or rough surfaces that can trap residues. Previous cleaning issues. Take into account any known historical cleaning challenges or issues associated with specific products, equipment, or surfaces. This helps address previously identified weaknesses in the cleaning process. Batch sizes and manufacturing scale. Select worst-case scenarios based on the largest batch sizes or scale of manufacturing. Larger volumes or equipment may present additional challenges for thorough cleaning. Cross-contamination potential. Assess the risk of cross-contamination between products. If there are products with significantly different therapeutic categories or allergenic properties, consider them as worst-case scenarios to ensure stringent cleaning measures. Regulatory requirements. Consider any specific regulatory requirements or guidelines regarding worst-case selection in cleaning validation. Different regulatory agencies may have specific recommendations or expectations for worst-case scenario identification. It is important to document the rationale for worst-case selection, including scientific justifications and risk assessments. By selecting appropriate worst-case scenarios, cleaning validation studies can effectively evaluate the cleaning process's capability to remove residues and ensure product quality and pay. Developing an effective equipment cleaning procedure is crucial in the pharmaceutical industry to ensure product safety, quality, and compliance with regulatory standards. Here are some tips to help you develop a robust equipment cleaning procedure. Understand equipment and product requirements. Gain a thorough understanding of the equipment and the specific product it will be used for. Consider the materials of construction, product compatibility, and any specific cleaning challenges associated with the equipment. Refer to regulatory guidelines. Consult regulatory guidelines, such as those provided by the FDA or other relevant authorities, for cleaning validation requirements and industry best practices. Ensure that your cleaning procedure aligns with these guidelines to maintain compliance. Identify critical cleaning steps. Identify the critical cleaning steps necessary to remove product residues, contaminants, and microorganisms effectively. Consider factors such as cleaning agents, cleaning tools, cleaning methods, like manual cleaning, automated cleaning, and the sequence of cleaning steps. Establish cleaning agent selection. Select appropriate cleaning agents based on the equipment and the types of residues or contaminants that need to be removed. Consider the compatibility of cleaning agents with the equipment surfaces, their effectiveness, and their safety for operators and the environment. Define acceptance criteria. Establish clear acceptance criteria for cleanliness to ensure that the equipment meets the required standards after cleaning. Define measurable parameters, such as residue limits or microbial counts, that indicate the cleanliness of the equipment. Determine cleaning methods. Determine the most suitable cleaning methods based on the equipment design, complexity, and the cleaning agents being used. This may include techniques such as manual scrubbing, rinsing, spraying, or using automated cleaning systems. Develop standard operating procedures. Document the cleaning procedure in a detailed standard operating procedure. Include step-by-step instructions, safety precautions, required equipment and materials, cleaning agent concentrations, contact times, and any specific considerations for different equipment parts. Validate the cleaning procedure. Perform cleaning validation studies to demonstrate the effectiveness of the cleaning procedure. This may involve conducting swab sampling, microbial testing, and visual inspections to ensure that the equipment is consistently cleaned to the required standards. Train personnel. Provide comprehensive training to personnel involved in the equipment cleaning process. Ensure that they understand the importance of following the cleaning procedure, proper use of cleaning agents and tools, safety protocols, and documentation requirements. Establish documentation and record keeping. Maintain thorough documentation and record keeping of all cleaning activities. This includes recording cleaning dates, personnel involved, cleaning agents used, cleaning results, deviations, and any corrective actions taken. By following these tips, you can develop an effective equipment cleaning procedure that ensures product safety, maintains regulatory compliance, and promotes consistent quality in pharmaceutical manufacturing processes. Today we will discuss the NOEL and MACO calculations in cleaning validation. NOEL is the No Observed Effect level of any pharmaceutical drug. It is determined to calculate the MACO, maximum allowable carryover, in cleaning validation. NOEL is the amount of drug in mg that does not have any effect on human health. NOEL is calculated by using lethal dose 50, LD50, of the drug. Lethal dose 50 is the amount of drug that kills the 50% population of the animals used in the test. NOEL equals LD50 multiplied by 70 kg, divided 2000. Where LD50 is lethal dose, 70 kg is average adult weight, 2000 is constant. For example, if any drug has LD50 331 mg, then its NOEL will be calculated as below. NOEL equals 331 multiplied by 70 divided by 2000 equals 23,170 divided by 2000 equals 11.58 mg. Now this value of NOEL is used to calculate the MACO, maximum allowable carryover. MACO equals NOEL multiplied by MBS divided by SF multiplied by TDD, where MBS is maximum batch size, SF is safety factor, 1000 for oral drugs, TDD is total daily dose of next product. For example, if total daily dose of the next product is 500 mg and batch size is 400 kg, then MACO can be calculated as below. MACO equals 11.58 mg multiplied by 400 million milligrams divided by 1000 multiplied by 500 mg. Here, 400 kg equals 400 million milligrams equals 9264 milligrams equals 9.264 grams. It shows that the drug with 331 mg per kilogram LD50 should not be carried over more than 9.264 grams in the next batch having 500 mg daily dose and 400 kg batch size. This criterion is used to calculate the cleaning validation limits. An effective swabbing technique is essential for accurate and reliable cleaning validation. By following best practices and guidelines, including proper swab material selection, pre-swab preparation, consistent swabbing motion, and careful swab collection and storage, pharmaceutical manufacturers can enhance the quality and integrity of their cleaning validation data. Today we will discuss sampling in cleaning validation. Cleaning validation plays a crucial role in the pharmaceutical industry to ensure that equipment, surfaces, and utensils used in manufacturing are effectively clean, preventing cross-contamination and maintaining product quality. A key component of the cleaning validation process is sampling, which involves the collection and analysis of samples to assess the level of residues or contaminants present. First of all, let us know the importance of sampling in cleaning validation. Sampling is essential in cleaning validation as it provides a representative assessment of the cleanliness of equipment and surfaces. It helps to identify any potential residues or contaminants that may remain after the cleaning process. By analyzing samples, pharmaceutical companies can evaluate the effectiveness of their cleaning procedures, identify areas for improvement, and ensure compliance with regulatory requirements. Now, let us know the key considerations for an effective sampling strategy. Sampling locations. Selecting appropriate sampling locations is crucial to obtain representative samples. These locations should include critical areas that are prone to contamination, such as equipment surfaces, joints, crevices, and areas that are difficult to clean. Consider worst-case scenarios and areas with the highest risk of residual contamination. Sampling techniques. Different sampling techniques can be employed, including swabbing, rinsing, direct surface sampling, or even equipment disassembly in some cases. Each technique has its advantages and limitations, and the choice of technique depends on factors such as the nature of the equipment, surface, or residue being sampled. Sample collection. Ensure proper collection techniques to minimize the risk of cross-contamination or sample degradation. Use clean and validated sampling materials, follow appropriate procedures for sample collection, and prevent exposure of samples to external contaminants during collection and transportation. Sample analysis. Analytical methods should be selected based on the nature of the residues or contaminants to be detected. Methods such as chromatography, like HPLC, GC, spectrophotometry, or specific tests for microbial contamination can be employed. Ensure that the selected methods are validated, accurate, and reliable. Sample size and frequency. Determine appropriate sample size and frequency based on statistical considerations, regulatory requirements, and the level of confidence required. Adequate sample size and frequency increase the likelihood of detecting potential residues and provide a more comprehensive assessment of the cleaning process. Documentation and record keeping. Proper documentation of the sampling strategy, including sampling locations, techniques, analysis methods, and results, is crucial for traceability and audit purposes. Maintain detailed records to demonstrate compliance with cleaning validation requirements. Sampling is a critical aspect of cleaning validation in the pharmaceutical industry. It allows for the assessment of residual contaminants, ensuring effective contamination control. By implementing a well-designed sampling strategy that considers key factors such as sampling locations, techniques, analysis methods, and documentation, pharmaceutical companies can enhance the reliability and accuracy of their cleaning validation process. This, in turn, promotes product quality, patient safety, and compliance with regulatory standards. Like this video, and share it with your friends and colleagues. Don't forget to subscribe to this channel and also hit the bell icon to get regular updates. Thanks for watching this video.